evening from wherever you're joining from. Thanks very much for dialing into today's webinar. We are really excited to have you on board. For some reason, my camera today just refused to turn on, but that will not be a big problem. Okay, so we're going to give it a couple more minutes as we wait for today's seminar presenter to join us. But I think I've seen a lot of participants that are familiar to a lot of us. I think we can use this time maybe to catch up a bit. I know for a lot of us it would have been a while since we last spoke to each other, so we can really use the time quite constructively to catch up whatever is happening elsewhere as we wait for the presenter to dial in. We're sorry about the delays, but we hope that it's going to work out with the presenter coming on board. So just to provide some insights, okay. So we were really privileged to have Professor Ette join us today. I can see he's already logged in. It's quite early. Professor Ette, can you confirm that you hear me?
Yes, I do.
Thank you. Thank you very much for joining us. We acknowledge it's quite early on your side. And there must be a lot of things going on, given that you're in the US at the moment. Do you have your presentation slides loaded on your side?
Yes, I have it with me.
Okay, I'll request that you share the slides. Then from my side, I'll introduce you, and then I'll hand over to you to present. I'm aware that there are a lot of people who want to ask quite a number of questions as people continue to join. So if we can reserve some time at the tail end of the presentation for questions and discussions, that would be amazing. The plan would be if you can do 30 minutes presentation, and then we'll leave the rest of the time for questions and discussions.
I think so.
Thank you. Thank you. I see the slides are loaded. Yes, thanks a lot. So I personally have known Professor Ette for now 10 years. I was checking. So 10 years ago, I attended the first lecture in Pharmacometrics, and that was given by Professor Ette alongside Joel Owen. And I've met Professor Ette in Uganda 4 times after the first time we met during the lecture. So I can assume that Uganda is certainly very close in heart to him. So Professor Ette is the president and the chief scientific officer of Anoixis Corporation, which is a pharmacometrics consultancy based in Boston in the US. He's a fellow of the American College of Clinical Pharmacology and Clinical Pharmacy, the Nigerian Academy of Pharmacy, and the American Association of Pharmaceutical Sciences. He has worked extensively in drug regulation, including working at the FDA for quite some time. And he won the inaugural Excellence Award in Review, in Excellency in Scientific Review for pharmacometrics related work. And just to stress this is actually one of the documents that gave rise to the current guidance documents that we often refer to for pharmacometrics. One thing is, if you've read a lot of pharmacometrics literature, you certainly might have come across his name somewhere. He has authored one of the worldwide most used books in pharmacometrics called Pharmacometrics, the Science of Quantitative Pharmacology. And there are several articles that have been published by him, including a lot that actually go towards the direction of developing new methodologies in pharmacometrics. Last but not the least, just last month, Professor Ette received the Shiner Bill Pharmacometrics Award, which is in this field one of the top most awards that scientists within the field of pharmacometrics can really receive. We are very glad to host you today. The title of today's talk is Pharmacometrics in Bioequivalence, Enabling Regulatory Environment. So this is one of the, I think, very important topics, and it goes in line with really the potential of using pharmacometrics in improving either drug regulatory approval in Africa or clinical use of drugs. So we are very excited to listen from you. And I do request all the listeners that if you have any question, please type it in the Q&A section. Later on, we'll also have the opportunity to engage in the discussions. Professor, I'll hand over to you, but first I'll request you to put your slides in presentation mode before we dive into the presentation. Thanks a lot.
Yeah, thank you, Francis. You asked me to speak on a topic that I don't really enjoy. Bioequivalence, but I do, do it when I need to. So what I thought I should do is kind of look at bioequivalence studies and then see how especially those in Uganda, I don't know the audience, the length and breadth of the countries, and I think also will apply to other African countries, what you can do to actually help the patients. And of course, when you do, job creation will be the offshoot of that. So I will also present an example of a bioequivalence study with a unique challenge, which I had to address. So I was outlined, talk about a little bit about what the pharmaceutical manufacturer season revenue is like, and then piggyback into a global bioequivalence studies market, and then talk about what I call bioequivalence knowledge creation, and under pharmacometrics knowledge creation, and of course a study as an example, and then from there do a summary. Move too fast. All right, so you can see here, this is worldwide revenue of pharma, and this is domestic to America. This is worldwide. So you can see that pharma is really productive and a lot in terms of revenue. But why do I bring this slide? It's just to show that in all this bioequivalence, play some part. All right, so here is U.S. bioequivalence studies market. All right, and you can see this is 2020. The amount of money spent there was 214, and it's been projected that as time goes on, you're gonna have this going up to going beyond the 679 that millions that was spent in 2022. In fact, the annual growth rate is approximately 8.4%. So then the question is, if this is the case, can we, in Africa, be able to benefit from this bioequivalence studies market, given the fact that the study is not complex, and the FDA does not really say, or even the EMEA where the study should be conducted. So now I put this here, which are therapeutic areas. You have oncology here, and you have other, oh, sorry. I gotta go back here. You have oncology and other areas that, I thought I'd taken this out. Sorry about that. Yeah, you have oncology here. You know, that might be complex because it's a patient study, but other diseases, you can do the studies in healthy volunteers. And that is the market, that's section of the market of therapeutic areas that studies can be done, you know, in Africa, I would say, Uganda and other countries. And of course, I'm sure the studies are being done in South Africa, but, you know, I'm focused on Uganda here and other African countries that I would call part of the emerging market. And so what can we do to try to see how we can change what is currently available or what is currently obtaining, rather than just be the recipients of what has already been produced? Can we be part of those producing, contributing to having a share in the market? All right, you know, I already gave you the global studies market by 2022. If you put that, add to that 8.3, 8.4% growth rate, and you can see that, you know, it's a lot of money being expended in that area and being made in that area. And the typical cost of bioequivalence in 2020 is $525,000 for small molecule drugs, and $525,000, and then $785,000 500 for ophthalmic solutions, emulsions rather. Now, the cost depends on the complexity of the formulation being studied. And several years ago, you know, average NDA submission as an estimated cost has been estimated to have contained about 3 to 6 in vivo bioequivalence studies. Now, if you assume that an NDA contains about one in vivo study, a bioequivalence study, and at that time the study was done several years ago, I couldn't find a new update. This was 1998. I couldn't find a new update. So we had about 350 to 600 BE studies annually. Now, with that trajectory that we saw there in the chart that I presented, you can see that this is increasing. And of course, FDA had said that companies should report all the bioequivalence studies that they do. So that tells you that there is something that can be, you know, can be explored here, and that we can be part of. Oops. Sorry, I'm moving too fast. That we can be part of. Oops. Okay. Now, we see the cost, and you know, after 2005, when a critical path for innovation was published, industry every year kind of encouraging industry to move into the model-based drug development. And this, of course, when I used to work at the agency at the Food and Drug Administration, we used to make fun of the bioequivalence folks that not much was done there apart from just playing around with AUC and CMAX. But now a lot has been happening in terms of research. In fact, they even have equivalence of a pharmacometric group in Office of Generic Drugs. And so what that means is that you know, there has to be a movement towards minimizing the occurrence of failed BE studies. And of course, the guidance even talks about virtual BE studies, bioequivalence studies. So with that in mind, we take a look at the biopharmaceutical classification. You know, this is based on solubility, permeability, and you can see, you know, class 1 and 3 here are highly soluble drugs, highly permeable and rapidly dissolving. So drug production. You don't have bioequivalent studies are not required for that. Basically, the solution can be used for the waivers. But bioequivalent studies are required for class 2 and class 4 drug products. Class 2 being low solubility, high permeability, class 4 being low solubility, low permeability. So those are this lower quadrant, the drugs in this lower quadrant are what are required, what the FDA requires that bioequivalent studies should be done. Okay, so now the question that I posed, I think I already said this before, is given the annual growth rate in the BA studies market, can Uganda and other African countries participate in it? And the other thing is that the regulatory authorities do not have a requirement where the studies should be performed. I think this is very important. They don't have a requirement as to where the studies should be performed. So studies can be performed in Uganda, Kenya, whatever, other countries in Africa, just like they've been performing in India. And so a question that Francis, you have to probably give us the answer to this, is BE trials performed in Uganda? Who approved the study protocols? Is the ethics committee centralized, or are they not centralized? Because if they're centralized, it makes it easy for a company to easily get a study, the IRB to approve the study and then get going. And, as a regulatory process, cumbersome or easy. Now, this is very important. The cost of BE trials in India is estimated to be 40 to 50% lower than in the US. All right. So if that is the case, I think that, in Uganda and other countries in Africa, it should be lower. That should put you, enable us in Africa to compete favorably. So you should have some kind of a comparative advantage. Of course, the regulatory environment has to be such that that can happen. So on this slide, I end with this, that there's a need to create value for patients in Uganda for any given drug in a national formulary, whether Uganda or other countries. Why am I saying that? For instance, almost all the small molecules that are already administered, if they're not class, not even class, but they are all it's supposed to have a high fat meal food effect study performed. Now, if I know that Uganda a little bit that I know, you are not on the high fat side, but you are meat eaters mostly, and so is the label that says take with food, is that relevant in the Ugandan environment? Or that could be different if the study was actually done, basically, because the food effect study is designed like a bioequivalent study, and the criteria is similar. Can that be done in Uganda to see whether actually if people can take whatever I said to be taken with food, it can be taken without food, because the high fat meal, which is a high stress test, does not apply. These are the kinds of things that, if taken into consideration, can, if there's a regulatory framework, then it should be able to create avenue for companies that want to register drugs in Uganda for marketing to be able to do at least a full effect study to make sure that the label on food effect is relevant to the society. And I think that, to me, is important, because it does create an avenue for the industry, clinical trial industry, to develop, and of course create job opportunities for pharmacometricians in the country and other countries. As you can see, that there's so many countries, so many companies, and the market is huge. So it's something that you need to interact with the regulatory agency to try to create that framework. So I put up here a case for pharmacometric knowledge creation in BE studies domain. And the objective here is to understand the likelihood of a new formulated innovator drug product being bioequivalent to the one used in a previous power single dose clinical trial during drug development before conducting a BA trial, kind of a long one. So basically, the question is, can a study actually be conducted, a BA study conducted, and the only information you have is data from a prior dose study. So the necessary ingredients for a BE knowledge base for a BE study is not there, but you're charged with the design of that study to come up with the numbers, sample size for the study to be performed. So when I talk about pharmacometrics knowledge creation, basically I'm saying that we can create knowledge by building upon current understanding of data, knowledge acquired by generating more information that is turned into knowledge. It entails the use of simulation models to synthesize data, supplement data for knowledge acquisition. Valid models, that is, when you want to synthesize data. That's what I'm going to be focused on, not the other part. Basically, you have data. You gotta discover the knowledge, or create a model, or develop a model from the data. Because once you develop a model, all that information in that data is summarized in that model. Then you can use that to then simulate data for what you want to do. So I'm putting some details here. The underlying basis here is model for further knowledge creation. And pharmacometric knowledge discovery is what we do always in analyzing pharmacometric data. We're trying to discover hidden knowledge in the data, because once we can discover that, we can synthesize that into a model. And with that model, then we can then use that to do other things. So knowledge discovery is a non-trivial process identifying valid, novel, potentially useful, and ultimate understanding patterns in data by characterizing data structure by means of a model. So data synthesis, of course, is it comprises modeling, where you have knowledge extraction, system characterization, and then, of course, you use that to do simulation. Basically, understand the system you're investigating and synthesize new data for further insight. Now, we do simulation all the time. I thought I should just remind us that simulation is a plan study, offers a powerful tool for evaluating and understand the consequence of study designs, which is what I'm focused on here. Is a useful tool to provide convincing objective evidence of the merits of a proposed study design analysis. That's the FDA 99 guidance that I wrote. So this is important because you want to understand things before you go there. Gone are the days that people do studies without knowing what is going to happen. Of course, they still do it today, and you say that, look, this is going to fail, and certain things should be done. They still don't want to do it, and then they fail, and then they come back. Of course, you have to tell them, I told you so. So here's the question, can a study be designed that successfully establish the B of a new formulation with a reference? And what is available is pharmacokinetic data for the reference formulation obtained from a conduct of a single dose escalation parallel study. This is actually something that I was confronted with when I worked at Vertex. At the time, we had just introduced clinical trial simulation into the drug development arena. And I was, of course, I always try to make friends with people that I work with. And I made friends with the biometrics director. I were able to convince the company to buy licensed software for clinical trial simulation. That was expensive then. Of course, things have changed now. And so now you have to do a BE study, come up with sample size, and all you have is a single dose escalation data parallel dose study. And so the pharmacometrics director, the biometrics director said, yeah, clinical pharmacology will provide a sample size. Now, for people that work in industry, that's the responsibility of biometrics. And of course, I had argued passionately to get a software, and I was trying to move the company into using PKPD as the basis, the pharmacometrics, the basis of drug development. And so now we had paid, some serious money then, to license the software, that we were to use. And the guy bailed out on me, and he was in a meeting. He said, well, and a clean fan will bring, of course, a clean fan, and they will bring provide the sample size. He was hiding under the guise that, okay, but BE is a phase 1 study, so yeah, that should be clean farm. But really, what it was running away from was that he did not have intra-subject variability data to be able to conduct a study. So what did I do? I had to perform a simulation study to determine sample size and power necessary to establish BE. I choose a sample size to yield approximately 90% power required to establish BE. And bioequivalent between the new formulation and the reference. Now, just as a side note, you do have BE studies, as I pointed out earlier, in drug development, where initial formulation, it wasn't that great. Now you have to switch. Of course, in bioequivalence, you're trying to compare to the proprietary product. Now, for the clinical trial, a sample size yielding at least 80% power is required. I use the word at least, but in simulations, I'm going to be using 90% power, and I will explain why. I use a 2x2 crossover study design, the optimal 2x2 crossover study design, and this was done in a faster state. Now, factors affecting bioequivalence, the study design is very important. The best designs are always the designs that balance variance across treatments. In this one that I'm going to describe, I use a 2x2 optimal 2x2 design. There are other designs in the literature, and the Ballam design, the Williams design, that actually is variance balance and enables you to use minimal subjects. So variability between and within subject variability is very important. Variability in physiological factors, and particularly the GI tract. For physiological based biopharmaceutical simulation, we're using PBPK model. That's important. Variability in active ingredient, pharmaceutical ingredient, and formulation, low solubility, low permeability, absorption window, dissolution, variable dissolution, GI degradation, first pass, and all that. So those things are important. And, then, of course, is when we think about design the study, we think about how we can optimize our study design to be able to, in sample size, to make sure that none of these things will knock us out, and make us knock us down and make the study fail. So for this simulation study, I did not use, now the example I'm presenting to you, this work was done in 1998. So I had to dig up, before I finally told Francis I could do this, I had to dig up, and I remember I did something way back. So this was done way back in 1998. As I said, it's a single dose study, a parallel dose study, the only data I had. And so to do this, I had to explore, sample size variability, inter-occasion variability, which is essentially within subject variability. I had to explore variability in Ka to account for those absorption GI factors, and variability in dose amount, variability in Ke, and variability in V. Basically, was a one compound model drug. So because I am not using PBPK, I'm using PK-based simulations. I have to make sure that I accommodate those factors of variability that we looked at, in this in Ka, for instance, for absorption Ka and dose amount, absorption Ka, GI, all that stuff falls into Ka, formulation variability and dose amount, and of course, interoccasion variability in Ke and apart from just between subject variability. So those are things that I factored in, I took into consideration. Now, the other thing that I mentioned is that I said to myself, the best thing for me to do to prevent failure, because the situation I was facing is, if the study failed, you say, clinical trial simulation doesn't work. Now, the setting was that we just started preaching clinical trial simulation from the FDA and move into industry. So now I gotta have to prove it. So here is simulation done, and then I can see here, the power and the sample size. I will vary from 20 to 30, and this is the intercellular variability in Ke, and you can see it increasing, up to 30. Basically, you can see there that, all right, let's see, for sample size of 24, thereabout, you will make a sample, a part of 80 will be okay, 90 will be, at 30 will be fine. So I wanted to bear that in mind. This is variability in Ka. In retrospect, this plot is not something that I would recommend, because it doesn't allow one to see the coverage of it. It's a slight increase here, but a slight increase, but it's not very, it seems to be very flat, so it's not a very informative plot. So the previous plot is what I would recommend. All right. Now here is the, oops, sorry, I moved too fast. Here is variability in dose. You can see, that you do have effect here. The SD in dose, basically is to account for variability from, because, when doses are produced, there is always some overage. So I'm accounting for that in case that the amount that the dose is not exact. So I have to account for the overage, the variability in that. And you can see that it does have some impact, on the power. Yeah, where is in that in Ke? Okay, so here is the importance of sample size. You can see that after all said and done, here is sample size of 24. All right, and I just showed how many studies that I took and summarized here, and you can see that in not every in some runs here that the sample size is oops, sorry. The sample size actually would not have, 24 would not work. Why am I? Sorry about that. You know, the sample size of this thing would not work. Actually, this plot is coming from the study after I've done. I think I put my slide ahead of it. This plot is actually after the study has been done, and I will show you, I think, some slides later, after jackknifing to see where a sample size of 30 will always make it or 24. Of course, if you have lower sample sizes, you can see that it doesn't make it at all. And you can see that at 24, some studies okay, some would not have worked. Yeah, here's the study results, and this is the real study results, which is, you know, my way of validating the BE study simulated study that I had performed. You can see that this is the reference, this is the test. Maybe this thing moves too fast. That's the reference, that's the test, and equally for this for AUC for CMAX. So basically, oops, the studies, sorry about that, the approach that I used to come up with the sample size actually, you know, work right for me. And a sample size of 30 is what we use for the recommended we use for the study. And here you're seeing the result. This is the reference. This is the test formulation, you know, for AUC. Same thing for CMAX. And you know, essentially we establish BE here. Now, what I wanted to find out is, oh no. This thing is too. What I wanted to find out is, okay, I had recommended a sample size of 30. What if I had done a sample size of 24? Would that work? So I did what I call a group at a time jackknife, and essentially the efficiency of a group at a time jackknife, which is taking a block of samples out at a time and re-estimating the sample size, and doing that in this case, that will be that will be 5 times. It performs similarly as taking out one subject at a time, one sample at a time, and re-estimating the sample size. You can see that out of all these, that in one occasion, the lower, we're looking at confidence level 90% should be 80 to 1 to at least 80 here, 120, 125, depending on what kind of drug. So you can see here that if I use 24, because each of these, you know, once you take out 6 is 24. In a situation where if it happened to be that, you know, it had some subject, 1 or 2 subjects, one subject maybe fell in here in the real study, then the study would have failed if we went for 24. So I say that basically that if when doing this, I used a 90% power. And I said, in reality, in going to the study, I said, okay, this sample size will give at least 80% power. At least 80% power. I didn't say 80%, I said at least. So you can't fault me for saying that. But to be on the safe side, it's best to run with 90% power. Because in that case, you can be sure that when you do your study, you'll be able to get, you know, establish bioequivalence, you know, with the sample size that you've chosen. So basically, I create knowledge about the BE that we're going to design in the absence of optimal information. And we performed a successful 30-man study, and we were able to eliminate BE failure, where that would have been the case if we had not performed this simulation study, given that we had no intra-subject variability. So, you know, that is a simple example that I give. I think the message here is that when faced with a challenge like this, you don't throw up your hands in the air and say, well, we can't do this, because, you know, when there's a will, there's a way. In this case, you know, the thought process was, okay, if I were to investigate, you know, variability both intra and between subject variability, wherever I can do that. In this case, it was one complemental model PK is for simulation, and then of course you can also do with 2 compartment. And then I should be able to, if I explore the universe of those 2 variables, of course, with sample size variation, I should be able to come up with a number that I have enough comfort to say, hey, listen, if we design a study, we're likely to succeed. And so that is what I did, and that's the result we got. And you can be sure that it did not, at least the company was encouraged that this was the right way to go, instead of saying that we have to abandon this platform and it doesn't work. And I think the other thing to remind us is, if we can get the regulatory agencies in our countries to buy into the fact that, you know, nutritional style and pattern is different, maybe they can think about that and think about opening up an avenue for clinical trial simulation. You know, for instance, in China, you cannot do a study in China and take data out of China. It must be analyzed by people in China. So with that, I think I end. I leave room for questions.
Thank you very much, Professor Ette, for quite an elaborate and insightful overview on pharmacometrics in the context of bioequivalence. Just to spell it out to the audience, please, if you have any questions, any comments, please type it in the Q&A tab of the Zoom link. We have quite a diverse audience, and we have about 14 minutes of questions. Please, I will invite you to type in your question once you have one. I'll use the privilege of being the co-host to ask a couple of questions first before the others come in, perhaps. Yeah.
Well, can I interrupt your privilege?
Yes, please.
Because there was a question here in the chat box.
Oh yes.
Do you want me to answer that first, or?
Please go ahead. I think I missed the question.
So yeah, the AT 120, the AT 120 125 is what is because the thing is both Cmax and AUC are log normally distributed. All right. So and it is assumed that, given that you have the log normal distribution, in that if you have that narrow band, you know, things can, because we're looking at average bioequivalence. And so again, number one, log normal distribution, and with average bioequivalence, we're trying to make sure that on average, at the end of the day, you end up with reasonable variability that doesn't give you things that are too wide. Now, if it comes to highly variable drugs, the study design is different. You have to use a design where the test is bracketed with reference. So you always have to repeat the reference more than the test, or in some cases, both of them, because of the high variability. Now, what I present really is not high variability in highly variable drugs, where the intracellular variability goes to 30%, you know, at the most. So that's what that is. Of course, for for non-linearity, and again, it will depend on whether it is highly variable or not. That will determine, the study design. All right.
Okay, thank you. I hope that was clear, and I think this is a very common thing that people struggle to understand with these bands, because if you don't take in the concept of normal distribution, then you miss the point. So,
And by the way, everything analysis with using log normal data, not log transform data, not untransformed, because we're looking at geometric ratio.
So the first question I have is, I think you've spelled it out that now we have a pharmacometric focused bioequivalence group at the FDA. And of course, as we try to really advocate for the possibilities of using pharmacometrics within this space, I think one of the key things that come out is to rely on, examples of where this is really done. I remember we had a discussion with the NDA at a certain point last year, and we didn't really have a very clear picture of where to put pharmacometrics. We know broadly how the field can work, but outside the general setting of conducting, clinical studies, it wasn't so clear how it fits in. Out of the FDA, you mentioned China and maybe Japan. Do they actually have frameworks where they directly use pharmacometrics in bioequivalence studies at the moment?
I mean, the FDA guidance makes for even virtual bioequivalence studies. Virtual. Kind of study that I showed. It makes for that. I think that, and you and I have discussed this over the years. That's why I put that full effect study thing in there. When we have the regulatory authorities understand that the diet in Uganda and maybe East Africa, if I can use that, is very different from America. I have been to Uganda. I haven't seen people who are massive in size. Right? So I mean, the forgot the number of calories. The high fat meal, forgot how much egg and all that is a heavy duty cholesterol, high cholesterol meal. All right, and that's not what is consumed in most of Africa. And so my thought is that if we get the NDA to buy into having a food effect study as a component of registration of drugs, you create an industry for clinical trial for BE. And then, of course, the next thing is, okay, we have this data. Who's going to analyze it? All right. Then that's where when you come in. But if there is no buy-in from a regulatory authority, they have not yet thought about how they can create a framework to employ people that are produced in the schools. That becomes a challenge, all right? Because there's no reason why you can't compete favorably with India, for instance, at a cheaper this thing. And in India, they do the study and do the analysis, and then send the results to the companies in America. So that can, that should also happen in Uganda and other countries if we're serious, right? Because if you start from there, then you create the clinical trial industry becomes one that can develop to cover other disease areas. Now you are in business because you now have more things that can be done, and then they can offer services that, look, we can also provide you with pharmacometric service, for instance.
Yeah.
So it's really making the cell, you know, because if you have at least from why I try to go for a low hanging fruit of food effect study, because that can now piggyback. Remember, I said that the guidance is provide for the fact that you can conduct this studies in any country. It didn't say it has to be America or Europe. If anything, pharmaceutical companies want where they can get cheap work, good work, paid for cheaply. And the cost of enrolling a student, a healthy volunteer in a study will be much cheaper than doing it here in the States.
Yeah, so it has a lot to do with actually creating that awareness and also showcasing that we have the capability locally across Africa really to do this kind of studies. I am aware that Dr. Mwebase, who is on the call, has done some food effect studies a couple of years ago, and she would really attest to where this benefit could come in. There's a hand up, but maybe let me just slide in this one question before I take the hand. So in the sort of the design of the bioequivalence study, you showed how you can use maybe a prior model that has been developed and then it optimizes the way you develop the study.
Yes.
Now, one thing that I see as a possible limitation is if the drug has not widely been studied, for example, in the local context, then probably you don't have a model that has representative parameters within the local context. So,
No, I mean, yeah, go ahead, finish.
So I am wondering how you would tailor a model developed from elsewhere and use it in the local context to inform really how you do a bioequivalence study with, you know, probably, how would you account for that difference?
That's a good question. That's why when you develop a model, you say that, you know, you read one of my papers called model appropriateness. We develop models for either descriptive or predictive purposes. And when a model is predictive, we're saying that the model is generalizable or transportable. That is, that a model developed in Boston can be used in Uganda. Now, you talk about the population. Generally, you know, unless it's a question of where there was maybe a population where there was some polymorphism. But here's the deal. You know, to be able to establish bioequivalence, you want to minimize variability. And so, you know, you can say that, all right, we're going to establish bioequivalence and maybe use the extreme poor metabolizers and basically recruit poor metabolizers so that you minimize the variability. Now, what I did there, without going into too much detail, was test, you know, variability in different aspects, variability in KE, variability in V, you know, and between and within subject, and vary. See, once you vary those things, you'll be able to see which one has the most influence. Of course, it's not that all these things are synergistic. They probably, you know, could be additive. And so you want to look at all those variables and then come to your conclusion. But if you have a model that is predictive, that's why we try to look at, you know, predictive performance of the model. It tells you the model is generalizable. Otherwise, it is not predictive, right? So you must have a predictive model to now say, okay, I got that, which is what I did there. Take a predictive model and then turn around and run the simulation. If I remember correctly, I think the first study was done in is it Netherlands, and then we did the BE in the US. So so that's the essence of making sure that you do have a predictive model. We say that your model is generalizable and transportable, and therefore you can use it. Now you have to play around with the different sources of variability, and that will now help you to come up with a solution. As I said, it's better to shoot for 90% power okay, in your simulations. And then in your study, your protocol, you say at least 80% power. All right. You're protecting yourself against failure.
Thank you very much. I think we have time for one more question. Martin, can you keep it short, please? Martin?
Hello, you get me?
Yes, yes, go ahead, please.
Thank you so much, Professor. Though Francis has preempted my question, I'm actually doing a study on metformin food effects, a PBPK model to predict food effects on absorption of metformin. However, I've struggled to get population data relevant to Uganda or Africa. So I was also asking about the importance of my model, but Professor has answered it quite well that maybe I should target a predictive model that can be used in different settings. So I also wanted to ask if IVC data can be used in place of bioequivalence studies, for example.
Physical, that's a different this thing, a different and that's mostly sustainability and all that kind of stuff. But when you talk about the bioequivalence, which is in this case a narrow limited to immediate release, this thing, you don't know replacement as such. Now let me go back to your PBPK model. I mean, the thing about PBPK, you better make sure you know you have your inputs, you get your inputs right. I think unless you have GastroPlus, which got all those things that you can use, but if you're trying to do this from first principles, it's going to be a challenge. That's why, and of course with PBPK, so many things are assumed to be true, with certainty rather, that are not certain. And so I try to stay away from PBPK because of that, unless I am forced to. I don't have a solution, other solution, then I can do it. Because, most of the things, the inputs into PBPK that are assumed to be certain, if you know how those studies are conducted to find those partition coefficients and all that stuff, they are not done with the rigor of normal studies. The people in Discovery just kind of by the fly, and so people use that, and then they say, okay, we did this, and they come up with twofold. I'm against twofold because it's a smooth way or smart way of reducing 200% off target to tell me that it is close. I mean, give me a break.
Sure.
You are 200% off target, you're off. I cannot recommend that to anybody. And so, unless you have those things that will give you that comfort level, I would have, I wouldn't, the way I like to be confident about what I present or what I recommend. So I wouldn't go that route unless you're confident that maybe you want to do so. Then check out GastroPlus, which has most of these inputs that you can easily manipulate. But if you're doing it from first principles, you have to find the data. And if you find the data, don't just, one of the things with PBPK, find that we assume certain population values, and it may not be relevant to your data. Okay? I did some PBPK where the flow, organ flow was certain number from literature, and I put it didn't work, and I had to estimate organ flow from my data before I got a good fit. You see, so but people will say, well, I got it from a literature. And I challenged one lady one time who said that, she did all this stuff, and people say, oh, she's very good on that. I said, you're so certain about uncertainty that you don't account for uncertainty. She said, she asked me, what did you say? I said, you're so certain about uncertainty, you don't account for uncertainty. That's the challenge with PBPK. And I think that sometimes you have to allow your data, even though you assume some parameters in literature, see whether those parameters actually can describe your model or not, your data or not. If not, then you have to allow it to estimate it, and then you can do this thing. But for your food effect, unless you're doing something for publication to show that you're smart, I would say that you can easily do use the approach that I used, and I think you'll be better off.
Thank you so much, Professor.
Mm-hmm.
Yes, thanks very much. Unfortunately, we've run out of time. I want to thank very much Professor Ette for sharing with us insights into this topic. You mentioned at the start that this is not the topic you want to talk about the most. Be sure that I'll get back to you on another topic that probably you like to talk about a little more.
Well,
I want to thank very much the audience for the active participation and the discussions. Professor, probably you're not aware, but there are a lot of enthusiastic quantitative pharmacologists on this call from across Uganda and actually across Africa. So next time when you're passing by Uganda, we should organize something. I would like to thank very much waking up in the morning and giving us this knowledge. We'll continue the discussion on things that you have advised us on. Finally, I would thank very much my co-host, Professor Katrina, and all the other members of the group, as well as our technical advisor on this call, that's Gabe from Pharmacometric Africa. Thank you very much, Prof. Have a good day over there. Bye-bye.
Thank you, and have a great day. God bless you all.
Thanks. Bye-bye.